This won't replace a formal training session if you're looking to get started on EM, but we'll give you the background and baseline understanding to be able to either A, tell whether this tool is appropriate for your research, or B, to understand and make sense of why researchers in your field use it and how to better interpret their data. All right, here we go. I'll start with some background information, which will essentially pick up where the limitations of light microscopy begin. Then I'll lead you through the basics of EM, including the physics and history behind these incredibly powerful machines and some of their common applications. Next, we'll cover some general use principles to give you an idea of how these microscopes are used. And finally, I'll go over some of the excellent resources available here at VCU. Now, we begin by understanding that how we as scientists visualize the tiniest aspects of our world. Since around the 16th century, this has been accomplished with light microscopes, with which you are undoubtedly familiar. The basic principle behind these microscopes is that we use light waves, which bounce off or pass through a subject to give us information about that subject. An advanced application of this principle, with which you're likely already familiar, is confocal microscopy, where light is filtered and passed through a pinhole to a sample, and the resulting light is filtered again and then collected. The same type of process will be utilized in EM, but we'll use electrons instead of photons. The resulting images from this light-based process are among the most striking we can create. On the left here is an image of cultured kidney cells, which I've stained to show the nucleus and cell-cell junctions. And on the right is an image from human atrial tissue, stained to show the overlap between gap junctions and sodium channels. This light-based imaging can be an extremely powerful tool, but is limited primarily by scale. As you can see, these images are taken on the microscale, but there is so much more information to gain orders of magnitude smaller than what these images can capture. So what happens when we want to look at something that's even smaller than the wavelength of light itself? Does the light simply miss? Well, not really. We just lose the ability to determine exactly where that sample is located. What we run into is known as the diffraction limit, which describes exactly how far apart we can detect individual points of light. This is determined by the wavelength of light, the numerator, and the refractive index of the system, the denominator. For green light with a wavelength of around 500 nanometers, this limit is about 250 nanometers. So this means that the closest 2 points can be and still be identified as distinct from each other is on the order of hundreds of nanometers. We can improve on this limit a bit with advanced super resolution light microscopy techniques, but only to an extent. So let's look at an example which I think we're all unfortunately familiar, and that's a coronavirus. We can also consider the space between adjacent cells. Now, this virus has a diameter of about 80 nanometers, while in the heart, cells are spaced within about 200 nanometers from each other. These values are well below, on the order of magnitude below what we're able to effectively characterize with current light microscopy techniques. Enter electron microscopy, which uses beams of electrons instead of visible light. If we remember from before, the diffraction limit is dependent on wavelength. When we calculate wavelength, we know that it is inversely related to mass. So wavelength and mass are inversely related. So since the mass of an electron is much larger than the mass of a photon, which carries visible light, the wavelength of a beam of electrons is much lower than that of visible light. All this means that the diffraction limit of an electron beam, which still exists, is orders of magnitude smaller than that of visible light, down to fractions of a nanometer. Electron microscopy began in the first half of the 20th century, which is still impressively early considering the technology and materials available at the time. They didn't even have computers, but they could still use electron microscopy. It began with the invention of the electromagnetic lens, a magnetic coil, which allows for the bending and direction of an electron beam, just as we use lenses to focus light in a light microscope. Shortly thereafter, German engineer Max Knoll and physicist Ernst Röska are credited for inventing the first electron microscope. Röska would go on to win the 1986 Nobel Prize in Physics for his work on electron microscopy. Dr. Martin would go on a few years later in Belgium to devise the first process for imaging biological specimens using an electron microscope. In Brussels. And those core techniques of using extremely thin sections, enhancing contrast with compounds like osmium tetroxide, and using copper mesh grids to support the sections are still used today. 70 years ago, his techniques for imaging biological samples are still used. Before we're even out of the 30s, Siemens had begun production on the first commercially available electron microscope. And since then, as technology has improved, particularly in creating more powerful electron guns and better electromagnetic lenses, we've seen great strides made in improving the resolution of electron microscopy down to about 1/20th of a nanometer with modern equipment. Today, you'll typically see two types of electron microscopes. The first is a transmission electron microscope, which Drs. Ruska and Knoll pioneered. A TEM uses an electron gun as a source of the beam, accelerates this beam through an anode at high voltage, through a series of lenses, through the sample, and collected onto a screen or camera at the bottom of the column. It's important to note that this entire process occurs in a vacuum, as even the presence of air particles in the column would be enough to deflect those electrons and distort the image. Next, there's also a scanning electron microscope, or SEM. Just like with the TEM, this machine uses an electron gun to generate the beam and accelerate it through a high voltage anode. However, instead of passing through the sample, the SEM uses scanning coils to focus and direct the beam, which then strikes a smaller piece of the surface of the sample. Backscattered or secondary electrons can then be collected, and that information can be used to recreate an image. The main strength of a TEM is its incredibly high magnification, as it is possible to get magnifications of up to 50 million times. No other microscope on the planet can get such high magnification. It can almost also image on a focal plane beneath the surface of a sample, similar to a confocal light microscope. And the resulting image is a 2D projection captured on that screen or camera at the bottom of the column. The main drawback is that the samples have to be extremely thin, less than about 100 nanometers or so that the electron beam can pass through the sample. The result is, again, an extremely powerful tool. Starting from magnification on the order of what we saw with confocal microscopy, this is an image of human atrial tissue. I was studying the structure of how the space between adjacent cardiomyocytes in patients with and without atrial fibrillation could affect things, and needed to be able to see at a much higher resolution than what I could get with light microscopy. So we can zoom in and start to make out some detail of this junctional region, where the darker smudges are more electron-dense areas, that is, areas with more proteins such as desmosomes or adherent junctions. Zooming in further, I can now start to identify the distinct regions I was looking for, gap junctions in the adjacent paranexus, which I could then characterize for my study. The main takeaway here is that TEM gives extremely high magnification cross sections of a given sample, whether that's within a cell-cell junction, where you can even see the characteristic gap, which gives gap junctions their name, or to identify spike proteins on the novel coronavirus. Another application of TEM is Cryo-EM. It uses the same system as a regular TEM, but utilizes a frozen sample, which can stand up to the vacuum of the column without having to be completely dehydrated. This technique allows for the characterization of individual proteins or other biomolecules. It is typically done at a lower voltage than other TEM applications, and is typically an alternative to X-ray crystallography or nuclear magnetic resonance, as it allows for greater flexibility of sample preparation. And finally, this technique won a Nobel Prize in 2017 for the high resolution determination of biomolecules. A great example of this application is the BRCA1 protein, mutations which have been linked to breast cancer. Excuse me, this particular example is from the work of a friend of mine, Dr. Cameron Morano, who's now at Penn State. And the way this works is a whole bunch of the protein of interest are suspended in a solution at random orientations and imaged. A computer then aligns all the similar orientations into groups and uses those groups to recreate a three-dimensional structure, as can be seen in the bottom left. You might notice with the scale bar that this protein is about 10 to 12 nanometers in length. And we're able to get incredible precision into its shape and density and how it changes with gene mutation to lead to disease. So Cryo-EM is an extremely powerful tool for the study of biomolecules. I'd like to also mention one more technique commonly used with TEM, but can certainly be applied elsewhere, and that's immuno-labeling. This is probably closest to what you've been used to before if you're familiar with light microscopy. We use this to determine where exactly specific proteins reside within the ultrastructure of a sample. Just like with immunofluorescence, we use a primary antibody to bind to the protein of interest, and then a secondary antibody with a sphere of a very electron-dense element, such as gold, attached. These electron-dense spheres stand out in high contrast to the surrounding tissue. For example, here's an image showing where connexin 43 is located in a cardiac sample. The investigator saw that not only did it show up in gap junctions, or expected, but also in some other structures as well. Finally, we can image different samples simultaneously. Instead of using different colored fluorophores, like with light microscopy, we actually can use larger or smaller gold spheres to differentiate between proteins of interest. Next, we'll move on to SEM, where again, we'll be scanning the surface of the sample down to a depth of maybe a couple of microns. The image is generated from backscattered or secondary electrons. Those are electrons that either directly bounce off of the sample from the electron beam or that are knocked off by the electron beam. The resulting image is a 3D reconstruction and has the advantage of not needing as involved a sample preparation process as with TEM, as the electron beam does not need to pass all the way through the sample, it just needs to bounce off. The main drawback compared to the TEM is that its resolution is limited to about 10 nanometers, which is about 2 orders of magnitude less than a transmission electron microscope. Even at that resolution, it's still possible to get some absolutely striking images. On the left, just for some consistency, is an image of cardiac tissue where you can see the sarcomeres within and intercalated discs between each cell. On the right, from the Santa Barbara Museum of Natural History, is an image of the head of a beetle. And on the SEM, it's able to capture in 3 dimensions the stunning clarity of the eye, mandible, and even the hairs on the beetle's head and antennae. So just to recap, TEM allows you to get extremely high magnification images of the cross-section of a sample, while SEM allows for a 3D rendering of the surface of the sample. It really depends on what you need for your own application. With that said, let's transition now into some general use principles of electron microscopy. Again, this won't replace a formal training session, but it should give you enough information to understand what you're looking at when you see EM images in literature, or to have a good foundation if you're looking to get into these techniques yourself. First, rule number 1 is don't break the equipment. These instruments are extremely delicate and expensive to maintain, and you should only use the equipment for which you have been specifically trained. I have about 5 years of EM experience at this point, and I wouldn't dream of walking into a new facility and just hopping on the microscope without going through a thorough training. Each machine is going to be slightly different. The big thing to remember is that with a couple of exceptions, all electron microscopes operate in a vacuum, and that creates immense pressures that can damage the equipment and your sample if you aren't careful. The good news is that everyone who operates these machines is eager to answer your questions. They're both excited to see what immensely cool things you'll produce with their equipment, and they're equally passionate about you not breaking said equipment. Finally, it's critical that you understand what's needed for your application. This can mean everything from equipment. Do you need a TEM, SEM, should your sample be frozen, lead stained, immunostained, etc., to the voltage of the electron beam you're going to shoot to or through your sample, and how your sample is prepared. Again, this is where there's an entire community, both here at VCU and beyond, of experts willing and eager to answer your questions. Now, before you can get to the exciting part of seeing your sample at extreme magnification, first must be prepared to survive in an electron microscope. This is probably the most critical part when it comes to interpreting the images you'll see, as this is where things can really go wrong, and it takes an experienced observer to really be able to determine whether what you're seeing is part of your sample or an artifact associated with the electron microscopy process. The main idea here is that the sample will be subjected to a high voltage electron beam in a vacuum, and therefore, with a couple of exceptions, you really can't image living samples. To survive in this environment, the samples must be fixed to survive contact with the beam, typically in an aldehyde solution, such as glutaraldehyde or formaldehyde, which forms cross-links between the proteins in the sample, allowing them to remain rigid in the path of the electron beam. And the samples must also be dehydrated, as in a vacuum, water boils at room temperature. That boiling water will literally rip your samples apart. This is typically done with, say, ethanol or acetone. From there, the process for TEM and SEM differ. As for TEM, the sample must be embedded in a rigid resin to allow it to be sectioned in extremely thin slices, which then allows for the electron beam to pass completely through it. These sections are then often stained with a heavy metal such as lead to enhance the contrast. For SEM, there is no thin slicing required, as the electron beam won't pass through the sample, but the sample is often coated with a very thin layer of a heavy metal such as gold in a process called sputter coating to prevent static charges from building up on the sample, which allows those electrons to scatter and be picked up by the SEM sensors. Once the sample is prepped, it's time to actually put it in the microscope. The first time you walk into an EM room, it's probably pretty intimidating, as there are all these knobs along the column. There's a loud pump to pull the vacuum, but it's really a straightforward process. The only part you'll even be touching on the column as a novice user is the sample holder. Now, this is an absolutely critical piece of equipment, as this is where you'll be interacting with the vacuum chamber. So just remember to be extremely careful, follow the facility's protocols exactly, and you'll avoid damaging the microscope. Likewise, on the control panels, you'll probably only be interacting with a few knobs until you really know what you're doing. This is because there are really two critical parameters when operating an electron microscope: the beam intensity and the magnification. This is how you control the beam itself, and in doing so, create the stunning images we've covered. To do that, you only need to use two knobs, the brightness and magnification. There's also a focus control to make sure you get the best image possible. The reason beam control is so important gets back to the idea that you're shooting a high voltage beam either to or through a sample. While most of the time, all the energy of this beam will be nice and spread out, it's possible to increase the beam intensity to a much finer point, and you actually can punch holes in your sample, which I've definitely done, or even damage the camera below on a TEM if you aren't careful. So one of the most important things to keep in mind is to keep that beam nice and spread out over the sample. Magnification is also super intuitive. Here's an image at about 1000x magnification, and I'll use this to navigate my way along the sample until I find a region that looks interesting. I can then zoom in this case to about 30,000 times, and look in greater detail. Finally, I'll zoom in a bit more to about 100,000 times magnification and have all the resolution I need for my structural analyses. The last aspect of getting a good electron micrograph is focus. Remember that biology never occurs in a perfectly flat plane, and at such extreme magnification, even the slight nanometer length variations in sample thickness can allow the structure you're looking at to drift out of plane. Again, at the microscopy core, this is controlled by a single knob and can really be the difference between a decent image and a great one. With proper focus, you're even able to make out the distinct lipid layers of the cell membrane. And when I'm looking at gap junctions, I can even quantify the width of that characteristic gap from a well-focused electron micrograph. Now you may be wondering who you can talk to about all the questions you might have and who would be able to train you to take such excellent images. Well, we have some truly amazing core resources available to us at VCU, and I'd like to highlight two facilities in particular: the Nanomaterials Core Characterization Facility, or NCC, and the Microscopy Shared Resource, aka the Microscopy Core. The NCC is located on the Monroe Park campus, and the Microscopy Core is in Sanger Hall on the MCV campus. To get trained on a particular microscope, or to have any of your microscopy questions answered, you can always email ncc@vcu.edu to get in touch with the NCC, or email Dr. Titus Bernas, who leads the Microscopy Core. I've had absolutely outstanding experiences with both facilities. Just to highlight some of the capabilities of the NCC, this is a slide from one of the recent updates showing their atomic resolution imaging, where you can see on the left and in the middle, TEM images with single-digit nanometer resolution and elemental mapping of a material sample on the right. While I focus on biological samples so far, it's important to remember that electron microscopy is a critical tool in the material science field as well, for everything from nanotherapeutics to electronics. And the NCC is set up to handle various types of both transmission and scanning EM, and can help you sputter coat SEM samples as well. When it comes to TEM sample prep, the Microscopy Core is the place to go. One of the best aspects of having dedicated microscopy personnel on campus is that their expertise developed over their entire careers is available to you even as a microscopy novice. An excellent example of this is an experience I recently had with the Microscopy Core in imaging some induced pluripotent stem cell derived cardiomyocytes. Although I was experienced in imaging tissue samples, cell monolayers were completely new to me, and the core had limited experience with this particular cell type as well. We followed essentially a standard protocol, and the result when I put in the TEM is up here on the left. Now you can kind of see where the mitochondria are. They're kind of those like little worm-looking structures, maybe an annular junction on the top right, but most of the cell structure looks like it's been washed out. If I was doing this alone, I would have spent untold weeks trying to troubleshoot and figure out what was going on, but instead, the Microscopy Core tweaked the dehydration protocol slightly, and voila. The very next try, I had exactly what I was looking for. These are the exact same type of cells as before, but here you can see the two nuclei, those little oval structures, and even the arrangement of muscle fibers in the immature cardiomyocytes. My project would have been held up for weeks, if not months, to get the same result, but this just took a few days and I was good to go. I really can't stress enough how valuable this is to have these resources available to us. I just let them know I'll be delivering an example. They give me a fixative solution, I drop it off, and within typically a couple of weeks, I'm on the scope imaging. And again, they're always happy and excited to iterate and help you get exactly the image you're looking for and make sure that the structures that you're really interested in are preserved and contrasted properly. And it's a really straightforward and pretty exciting process. With that, just to sum up, electron microscopy is among the most powerful imaging tools available to us as scientists. No other technique can achieve the magnification and resolution of an electron microscope. Just be sure to tailor your approach to the specific needs of your project, and remember there are always dedicated microscopy personnel on campus who are happy to help. Along those lines, always ask questions, whether it's sample prep or even basic microscope controls. Again, the folks who run these facilities are happy to help. You will never sound dumb when asking a question, but you will always look and feel quite dumb when you damage the scope and it's down for weeks while we wait for parts and a technician. So always ask questions. That said, training and use of these machines is surprisingly straightforward. A training session is typically a few hours, and then you're good to go to start imaging. Finally, there is so much to explore at the nanoscale. I really can't encourage you enough to incorporate EM into your project. The first time I imaged cells and tissues at this scale, I was blown away by how much there was to explore and how oversimplified all the cartoons in our science textbooks are. I've seldom felt more relaxed and excited than when I'm sitting at the EM scrolling along a cell looking for interesting structures. There are virtually infinite research possibilities to probe at the very fundamental structures of life and materials. And again, I encourage you to check it out given the opportunity. At the very least, I hope I've given you a good introduction to electron microscopy and helped you to understand and interpret the next EM image you come across on paper. And with that, thank you so much for your time and attention, and I'd be happy to answer any questions.